everyone tends to get stuck on this very polarizing binary question of whether or not animal research should be completely abolished, or is animal research entirely justifiable because it benefits people? I don't think that's a very useful question, and it hasn't really gotten us anywhere.
I've been at MIT for just under 30 years. I came here as a graduate student, and having no idea at the time I would possibly work on this animal called the zebrafish that was becoming a new, interesting research organism. A lot of the earlier work in zebrafish was really looking at the development of the animal. Folks will use zebrafish as a model for many, many human diseases, including, say, liver disease, kidney disease, heart disease, investigating cancer, obesity, investigating alcohol sensitivity. It can be more efficient or easier to learn things in the fish, especially if you can study things while they're still embryos. Because the fish are rather transparent, you can see lots of things.
One of the cancers we study is a cancer called uveal melanoma. Pigmented cells are sitting within the eye or at the back of the eye, and they're the ones that get transformed. You cannot study cancer efficiently only doing it in vitro in a dish. You really have to study cancer in the context of a whole organism. That's actually one of the beauties of zebrafish, is that we can really manipulate the genetic makeup very easily. You can actually inject into one cell and make genetic changes that would then be propagated through all of the cells and the whole organism. So sometimes it takes a year to develop enough mice of the appropriate genotypes to have statistical significance in our experiment. And with zebrafish, we can often breed just for a couple of weeks, and we have enough animals for that study.
Previously, it was thought that we had to keep animals in the most sterile and controlled environments as possible, because otherwise we couldn't interpret the results of experiments.
It's critical that animals are healthy and happy. So for example, in cancer, it's very clear that stress or infection with certain viruses or parasites can actually change tumor phenotypes.
My old boss used to like to call our fish facility Fish Ritz. We treat our fish extremely well. They have a lovely life. They are fed 3 times a day. We hatch brine shrimp. It winds up that having live food stimulates them socially, intellectually, and that's important for their behavior. And they're fed a diet mostly of that, but they also get some sort of commercial flakes. We check the variety of parameters of the water. Mostly the pH is the main thing we're looking at, but also temperature and salinity.
I don't think zebrafish are very cute. I think it'd be very difficult to work with zebrafish and also find them cute. I work on embryogenesis. It's this phase when the embryo goes from the one cell stage, which was just an egg and a sperm, and becomes a real fish. A typical day of me using the fish would be that in the evening, I'll put together a male and a female fish, and they will sit quietly all night together. They will start breeding, and eggs will be released from the female and be fertilized. I'll go down to the fish facility and collect these eggs that are newly fertilized. Zebrafish eggs are contained in something called a chorion, something akin to an eggshell. And if we want to image the animal, or we want to manipulate the animal, which is now contained in this transparent eggshell, we sort of need to maneuver a forceps into that eggshell and rip it off so that the animal can now develop, and that we can image it in a much better way. We euthanize animals by anesthetizing them first. We use a drug called tricaine, and it will anesthetize the animals so that they can't feel any pain, and then we put them into either an ice solution, which will euthanize them, or we put them into a bleach solution, which will euthanize them. I've used, over my time at MIT, roughly 1,000 zebrafish adult animals, maybe 10 to 15,000 embryos. I do a variety of biochemical purifications that are difficult and require a lot of material. My gosh, 15,000, Jesus Christ. Yeah, right, yeah, I know.
A lot of what we do, we could not do without the animals. Is the experiment critical? Can it only be done in animals? How few animals do you need? What are the appropriate ways that you can do it to minimize pain and suffering?
Part of the reason that many researchers chose to use fish as their models was the historical belief that they didn't feel pain the way that other kinds of animals do. But the problem is that welfare is not very well understood. So we're often working on very incomplete information. Even though fish do not have the same kind of organized nervous system that mammals do, they are still capable of feeling and perceiving pain.
The vast majority of people have a real sense of responsibility about using animals in their research and thinking about the contributions that they make. In fact, often I see in people's talks, at the end of their talk, they'll thank the mice or they'll thank the zebrafish because they've made a critical contribution in this experiment.